Hello, my name is Abigail, and I did my research project on the treatment of late-stage adenocarcinoma, which is just cancer in that GI tract. So treatment of late-stage GI cancer using genetically modified mesenchymal stem cells. What mesenchymal stem cells are is they're cells that line the hepatopancreatobiliary region, and they bring oxygen and nutrients to the intestinal area. And because of the delivery of oxygen and nutrients, the area is very conducive for tumor growth. So one common problem is that tumors will begin to grow here, and they can grow so rapidly because of the delivery of oxygen and nutrients, and cancer progresses very quickly here. And as I'll touch on later in the presentation, the rates of survival for these types of cancers is very, very low because it can grow so well in this particular area. So that's a little bit about what mesenchymal stem cells are. So I'm just going to give a brief overview of the topic that I researched, and then the PowerPoint goes into more depth and kind of explains things more thoroughly. So as I pull up the PowerPoint here, so this was a 2017 study that was done. So again, novel, recent genomics. That's the theme in genomics. It's very novel. So they took these stem cells and they inserted into these stem cells something called transgenes. And this gave these genetically modified mesenchymal stem cells the ability to migrate very deep into the tumors and kill these cells in the tumor. And now is when I'll get into how that happens. So you have the transgenes inserted into the MSC cells, the mesenchymal stem cells, and these modified stem cells can either do one of two things at this point. They can encode for a protein that has an inhibitory effect on the tumor, or they can encode for enzymes that will phosphorylate the non-toxic antiviral ganciclovir into a toxic form called ganciclovir triphosphate. So it's been phosphorylated. Then the herpes simplex virus thymidine kinase is used to initiate that phosphorylation. So try not to get caught up in the fact that it's the herpes simplex virus. That confused me a bit. The only purpose is that it's being used to initiate phosphorylation of ganciclovir into ganciclovir triphosphate. So once it has been phosphorylated, now you have this toxic ganciclovir triphosphate, and this toxic compound is very easily sent through the gap junctions, and it is ultimately taken up by the mesenchymal stem cells. So one important thing to note is that when these tumors are growing, they're constantly recruiting more mesenchymal stem cells to keep up with the increased demands for oxygen and nutrients. So now this tumor has just accepted these genetically modified mesenchymal stem cells. And the key to this whole thing is that these cells that have been genetically modified and activated using herpes simplex virus thymidine kinase, they have inside of them, they have programmed cellular death, which is another term for apoptosis. So these mesenchymal stem cells migrate deep into the tumor, and they seem like they're just normal cells, normal mesenchymal stem cells, but they are not. They have the ability to kill themselves, and they're called suicide genes. So once they are deep within the tumor, they begin to undergo apoptosis. How this happens is the toxic ganciclovir triphosphate inhibits DNA polymerase. So this means that the DNA is not going to be doing all of the replication, the unwinding of the DNA bend. It's not going to be doing all of these processes correctly. So ultimately, this is going to lead to apoptosis. And because these stem cells are so deep within the tumor now, they're dying, and this means that the tumor is getting decreased oxygen, decreased nutrients, and it's going to begin to shrink in size, or that is the ultimate goal of using these stem cells as a genetic treatment. So I spoke earlier how the survival rates for these pancreatic cancers or colorectal cancer, very low survival rates. So for patients with regionally advanced pancreatic cancer, the 5-year survival rate is between 13 and 14.2%. that's almost nothing. The 5-year survival rate for intrahepatic cholangiocarcinoma, this is a difficult one to pronounce, cholangiocellular, so intrahepatic cholangiocellular carcinoma, their survival rate is between 2.4 and 2.7%, even lower. And then for colorectal cancer, your initial survival rate is 71%, but once the cancer metastasizes, your survival rate drops down to 13%. So there's obviously an abundant and overwhelming need to find a way to treat these cancers. And the hope of using these genetically modified mesenchymal stem cells is, number one, our body recognizes that our body does not recognize them as foreign, so they can migrate into the tumor and they're accepted into that environment. And the goal is that as they undergo apoptosis, this will decrease the tumor size and ultimately either hold off the cancer. It can't cure it, but it can hold off the cancer. And something that the study found was that the median overall survival was 15.6 months, which was more favorable than if there had been no treatment. So just thinking about this logically, there are some problems that arise. When you give someone the injection of these modified mesenchymal stem cells in addition to ganciclovir, how do we know that these cells that will undergo apoptosis are going to migrate to the tumor and not another healthy part of the GI tract? So I'm sure that the scientists have thought of this before. In the articles that I read, there was no indication that they had been able to track where these cells were going, although the median survival rate was more favorable, so we're assuming that they went to the right place. But that's just something that I thought of, and that's why genomics and genetic treatment like this is so novel, because there are so many things that need to be worked out still. But using this type of treatment is very innovative. Using cells to migrate into tumors and ultimately undergo apoptosis, the prognosis could be very favorable, and I expect to see in the coming years a development of this and many other genomic processes that will lead to many more favorable prognoses for diseases.